Good afternoon, everyone. My name is Jin Yang. I'm from the University of West Ontario, Canada. Thank you for organizing committee inviting me to give this talk. The title of my talk is Study of Conductive Bacterial Nanowire, Fundamentals and Applications for both Environmental and Energy Sustainability. So this is the campus of our university. The University of West Ontario, also called the Western University, was founded in 1878. This is one of Canada's top research intensive universities. This is the outline of my talk. First, I will introduce the background of electric conductive bacterial nanowire, and then I will talk about our experiment result and discuss our result, and then finally I will talk about my perspective about applications of conductive bacterial nanowire for energy and the environment sustainability. The basic principle of biological energy generation is based on oxidation and reduction process involving electron transfer within cell and cross the cell membrane to the terminal electron acceptor. In the case of electron transfer outside of the cell, known as extracellular respiration or extracellular electron transfer. Bacteria can generate energy using diverse strategies. The fundamental principle of energy generation for metabolism is based on transfer of electron from electron donor with lower reduction potential, also known as reduction or oxidation or redox potential through an electron transport chain to terminal electron acceptor with a high reduction potential. The electron transport chain couples with the electron transfer to the transfer of protein across the cell membrane, producing an electrochemical protein gradient as a result of a series of redox reaction release chemical energy. For example, this is glucose is through this reaction to generate ATP, generate energy. So electron transfer is a fundamental mechanism for energy generation in any living systems. So what if the electron acceptor are poorly soluble or insoluble? How extracellular electron transfer is conducted? So in literature, people found three possible strategies for extracellular electron transfer. First is direct transfer using multi-stem cytochromins or electron transfer protein through this direct contact. Another mechanism is through the transfer through the organic molecules in solution by diffusion. The third mechanism is what we have been studying is electron transfer by cellular appendages, or here we call electrically conductive bacterial nanowire. This phenomenon was first discovered in 2005 and 2006 on dissimilatory metal-reducing bacteria. For example, on two types of bacteria, Geobacter and Shewanella MR1, people discovered these two types of bacteria can grow bacterial nanowires which are conductive. Let's look at this 2005 Nature paper. The authors use the conductive AFM to study the conductivity of bacterial nanowire. So on this figure, is this the topography of the bacterial nanowire, and this is their conductive AFM result showing the bacterial nanowire of Geobacter, which is conductive. However, all previous measurements were done across the thickness of the nanowire. So the question we want to answer first is, can electron be transported along the length of bacterial nanowire? So in this direction. So with our colleagues, we first culture the bacteria. We use the Shewanella MR1 as the example. After cell culture, we do characterization on the bacterial nanowire using a variety of characteristic methods, for example, SEM, TEM, optical microscope, AFM, STM. This is a image of SEM and TEM. Under SEM, you can see bacteria grow nanowires and form a network in the biofilm. And we also stain the bacterial nanowire and showing that bacterial nanowire is protein nanowire. Next, we repeated the 2006 PNAS paper's result, also do conductive AFM. This is the morphology of the bacterial nanowire. In our result, we also show the conductivity of bacterial nanowire across the cross-section of the nanowire. Here, for example, the brighter region is the nanowire showing its conductive. So next, we want to answer how electrons transport through the length of the bacterial nanowire. So the most straightforward method is to place two electrodes on the bacterial nanowire to measure the IV curves. For example, you place the electrode on the bacterial nanowire and then add a voltage and then measure current. Okay, so in reality, since the bacterial nanowire is not very long, so we cannot place big electrode, use a voltage meter or multimeter to do this measurement. Instead, we have to do this through E-beam deposition. So under SEM, we find the bacterial nanowire and then use the E-beam deposition to grow two electrodes to bridge the connection. And then we add voltage to measure the current. So we found the resistivity is around 1 ohm centimeter for wild type. We also did the measurement on flagellin deficient mutant. We also found the conductivity or resistivity of this type of the mutant, the bacterial nanowire, the nanowire of the mutant bacteria. So we also do control experiment. Since we do, people may argue the conductivity is due to the metal deposition in this E-beam deposition. So we do this control experiment. What we did is we cut the bacterial nanowire use R-beam and then make it open current. So there is no after we cut the bacterial nanowire, we don't get the current. Means the current indeed pass through the nanowire, not the E-beam deposition does not cause the conductivity in this measurement. And then, we place the 2 electrode in the bacterial nanowire to do the measurement. But ideally, we should do 4 probe measurement instead of 2 probe measurement to eliminate the effect of conductive resistance. Note that the bacterial nanowire is short. Placing 2 electrode is already very challenging. It's more difficult to place 4 electrode. So we choose another way to do this measurement. So we do conductive FM, and by move the FM probe along the length of the bacteria to measure the IV current at different distance from the electrode, or measure the resistance of the bacterial nanowire and the different lengths. And what we did, we make the gold micro grade, and then place the bacteria on the substrate. If we find there is a bacterial nanowire just get into contact with one electric gold pad, then we will do this measurement. Then we will choose that bacterial nanowire to do measurement. So for example, here we can find that one bacterial nanowire placed on a gold pad, then we can do the measurement. And what we do is we're still doing conductive FM. And then we measure the resistance along and different location along the bacterial nanowire, and then we can get this data point. If we extend this line, we fit the data and extend this line to the 0. The x-axis is the distance from the electrode. If we extend to x equals 0, means there's no its distance from the electrode is 0, means there's no conductive resistance. So means at this point the resistance we got is the conductive resistance between the conductive FM probe to the electrode. So we can characterize the conductive resistance is. Using this, then we can calculate the resistivity of the bacterial nanowire again. So still we get the very similar result, 1 ohm centimeter. So 2 independent methods, we get the same result, which confirm the resistivity is 1 ohm centimeter. So in this experiment, in this paper, we answered the first question: what's the resistivity along the bacterial nanowire? So our next question is, what's the conductive nature of bacterial nanowire? We did this experiment on on two substrate. When we did a similar experiment, conductive AFM experiment on two substrate. One substrate is gold, another substrate is HOPG. We found that on HOPG, we get this nonlinear IV curve. On gold substrate, the probe is coated with platinum, PT. So on gold substrate, we found this linear IV curve. So which indicate bacterial nanowire behave as a semiconductor. Because according to the theory of semiconductor physics, when there is a difference in energy between the Fermi level of a semiconductor and that of a conducting metal, charges must overcome an energy barrier called a Schottky barrier in order to pass across the interface. So this nonlinear IV curve indicate this is the nanowire behave as a semiconductor, and therefore a Schottky barrier exists and the nanowire and HOPG junction, owing to Fermi level mismatching. In fact, this nonlinear IV curve was also found in 2005 and 2006, 2005 Nature paper and 2006 Peanuts paper. So we fit the curve. We found that for when the girl use the gold substrate, the IV current is linear. So if we look at their the Fermi level of those materials, for platinum is 5.3 EV, for gold is 5.1 EV, HOPG is 4.6 EV. So the linear IV curve indicate we got the ohmic contact. The nonlinear IV curve indicate there is a Schottky, which is a Schottky contact. So which already indicate bacterial nanowire behave as a semiconductor. So we use the most traditional way to characterize this semiconductive property of the bacteria. To do that, we build the field effect transistor still by our nanofabrication. If we find the, on this gold silicon dioxide substrate, we use the E-beam deposition to grow the two electrode as a drain and the source electrode. And then we can add gate voltage and measure the current between these two electrodes. So this is the result. We found if we change the gate voltage, for example, if we increase the gate voltage, the current reduced. So which indicate the charge carrier is holes. Charge carriers are holes, and this bacterial nanowire is p-type like semiconductor. And we fit the curve. What we found the charge mobility is about 2.3, and which is very similar to P3HT. Then further, we characterize the elasticity of the bacterial nanowire, and we found the elasticity similar to most of the polymer. So we think this bacterial nanowire conductive as silicon, but flexible as elastomer. So which indicate this kind of bacterial nanowire can be used for organic electronics. Then next, we want to explore this bacterial nanowires application for energy and the environment. The technology we choose to demo this application is microbial fuel cell, because the microbial fuel cell can do can have two functions. While to do water treatment, decompose organic compound in wastewater, meantime generate electricity. So this type of microbial fuel cell have two advantages, two functions for both energy and environment applications. So prospective application of microbial fuel cell is maybe one day we can install such a microbial fuel cell in each individual house to treat the household wastewater, meantime generate electricity for a single room. So one of the great challenges in developing such microbial fuel cell is to get the efficient electrode. For example, to make the porous material, porous electrode, which can give you highest efficiency of the power generation, and the meantime to decompose the organic compound in wastewater. So this requires us to control the micro texture of the electrode very well. So how do we control the micro texture of the electrode? We use 3D printing. So first we use the 3D structure and coated with copper as an electrode, but we found the copper electrode degrade during the use of microbial fuel cell. So later, then we move to the carbon-based electrode. We still doing the process is like this. We do 3D printing and then do carbonization to convert the polymer electrode into a carbon electrode. By 3D printing, we can control the morphology, we can control the pore size, we can control the porosity of this type of electrode, and to achieve the best efficiency and the best environment for biofilm to grow. So we can do different characterization of the biofilm under different for different pore size, which we make such carbonized electrode with different pore size, and then we can study how bacteria grow on different electrode, and what's their for example, we calculate the thickness of the biofilm, and what's the coverage, and then also measure the conductivity of the biofilm. And later, then we were doing measure the energy, how much energy. For example, voltage. We measure the voltage and the power density of such microbial fuel cell for different pore size of electrode. So those are results, and here we would like to summarize in this for different pore size. And we do this measurement. We found that when the pore size is about 300 micron, the microbial fuel cell can achieve the highest efficiency in terms of power density and the voltage generated. And compares to the carbon closed electrode, the efficiency of 3D printed electrode is much higher. So to summarize, Shewanella bacteria nanowire are electrically conductive along the length with the resistivity and the order of 1 ohm centimeter. And this bacterial nanowire is a p-type semiconductor, and which also have very similar charge mobility to P3HT, and so which indicate maybe this bacterial nanowire can be a replacement to P3HT. And the Shewanella bacterial nanowire are conductive as silicon but soft as polymer, and which indicate its applications for organic electronics and flexible electronics. And microbial fuel cell, if can be used for wastewater treatment and power generation for both energy and environmental sustainability. So we can say in the future, maybe we should use more and more bioenergy and using bacteria to generate energy as our future power supply. Thank you for your time and attention.